Hello everyone, and welcome to today's The Scientist webinar. My name is Nathan Nee, Associate Science Editor for The Scientist, and I will be moderating our discussion today. Today, our speaker, Dr. LuZhe Sun, will discuss TGF-beta signaling during cancer progression and how researchers can target this pathway. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and our speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these questions as we can during our Q&A session. This webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. At this time, I would like to take this opportunity to thank our webinar sponsors. Sinobiological is an international reagent supplier and service provider, as well as a global leader in recombinant technology, specializing in recombinant protein production and antibody development. Sinobiological offers life science researchers high-quality recombinant proteins, antibodies, ELISA kits, cDNA clones, and one-stop CRO services to accelerate their research in a variety of fields, including cancer, immunology, drug discovery, and infectious disease. For more information, please visit www.sinobiological.com. Nanotemper's mission is to enable everyone to do science that matters by always pushing the limits. They focus on making tools for drug discovery and basic science research that address challenging characterizations of target molecules involved in cancer and other diseases. If you're facing challenges with affinity screening, molecular interactions, protein stability, or protein quality, they'd love to talk to you. www.nanotempertech.com. 10x Genomics builds solutions to interrogate biological systems at a resolution and scale that matches the complexity of biology. Their rapidly expanding suite of products, which include instruments, consumables, and software, have enabled customers to make fundamental discoveries across multiple research areas, including cancer, immunology, and neuroscience. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our handout section, located on the right side of your screen. You can access and download these documents at any time during the webinar. With that, let me introduce our speaker, Dr. LuZhe Sun. Dr. Sun received his PhD in physiology from Rutgers, the State University of New Jersey, and UMDNJ Robert Wood Johnson Medical School in 1990, and did his postdoctoral training at the Baylor College of Medicine. He is currently Professor of Cellular and Structural Biology, Dielman Endowed Chair in Oncology, and Associate Director for Translational Research at the Cancer Treatment and Research Center at the University of Texas Health Science Center. Sun researches molecular mechanisms of tumorigenesis and metastasis, and novel experimental therapeutics in various models of carcinoma. He co-discovered the tumor suppressor role of the TGF-beta type 2 receptor in hereditary non-polyposis colorectal cancer. He also pioneered the use of a soluble TGF-beta type 3 receptor and engineered a soluble chimeric TGF-beta receptor for the inhibition of cancer growth, angiogenesis, and metastasis in various preclinical models of human cancers. Currently, his laboratory investigates the effect of environmental compounds in aging on the function, genomic integrity, and transformation susceptibility of mammary stem and progenitor cells, as well as the potential molecular etiology of hepatocellular carcinoma in South Texas Latinos. His research has been supported by R29, R01, and P01 grants from the National Institutes of Health and grants from the Department of Defense, the Cancer Prevention and Research Institute of Texas, and the Clayton Foundation for Research. He has served as a charter member of the Cancer Biomarker Study Section and the Tumor Progression and Metastasis Study Section of the NIH, and the Subcommittee F Initial Review Group of the NCI. He has regularly participated in grant review panels for the NIH and DoD. Sun has published over 100 peer-reviewed research manuscripts, and he was elected to be an AAAS Fellow by the Council of the American Association for the Advancement of Science in 2015. Let's just check that everything is working from a technological standpoint and everything appears to be okay. Dr. Sun, the floor is yours.
Great. Thank you, Nathan. I assume everybody can hear me and can also see my slide. Again, I want to thank Nathan for the invitation for me to tell you, with this webinar, to tell you the development of some novel TGFβ traps over the years in my lab to really inhibit tumor progression and to enhance anti-cancer drug activity. So what I will do in the next 40 minutes or so is to first briefly introduce TGFβ and its signaling transduction, review with you the role of TGFβ pathway in tumor progression, and then tell you how we have utilized soluble TGFβ receptors as TGFβ traps, sometimes we call TGFβ inhibitors, for the inhibition of tumor progression. And I will also show you some data that indicate that the chemotherapeutic agents can actually stimulate TGFβ production and activate the TGFβ signaling. And finally, I will use a few slides to show you how we have used a novel TGFβ trap to enhance the anti-cancer drug activity. So TGFβ is transforming growth factor beta, and it is a polypeptide. The mature active TGFβ is actually a homodimer of 25 kilodalton, and it is synthesized and secreted in a latent form, in that the pro region of the TGFβ is still non-covalently associated with the mature active dimer. And TGFβ, the latent TGFβ requires activation to release this active TGFβ dimer for its signaling through its receptors. And that activation involves various kinds of extracellular matrix proteases, as well as integrins that TGFβ latent protein, binding protein will bind to. So TGFβ signals through two cell surface receptors called the type 1 receptor here and type 2 receptor here. So TGFβ is known to first bind to type 2 receptor, which recruits type 1 receptor, and both receptors are serine threonine kinases. So as type 1 receptor is recruited to this ligand R2 complex, then it's being activated by phosphorylation through the type 2 receptor. So the activated type 1 receptor then will phosphorate the intracellular mediators called Smad2 and SRIII, and the phosphorylation of Smad2 and SRIII then will interact and bind to this co-Smad called Smad4. And this trimeric Smad complex then translocate to the nucleus, act in the nucleus as transcription regulators or transcription factors to regulate the TGFβ signaling pathway targeted genes. Now, TGFβ has also been shown to signal through this Smad independent pathway by activating some of the MAP kinases here shown here, as well as the PI3K and AKT kinase. So both the Smad dependent pathway and Smad independent pathway will regulate gene expression and cellular function, as we will discuss subsequently. So TGFβ has been described as a double-edged sword in terms of its regulation of tumorigenesis. On one hand, TGFβ has been shown to act as a tumor suppressor. On the other hand, it has also been shown as a tumor promoter. So in Chinese, we call this as yin-yang effect. So TGFβ was initially discovered to be a growth inhibitor, and as shown here. So signaling through the receptor Smad, it has been shown to target multiple genes that are involved in the regulation of cell cycle here, including a number of cyclin-dependent kinase inhibitors, as shown here. So TGFβ increase their expression and decrease the expression of some of the growth promoters, such as CVIC and E2F. So on the other hand, the TGFβ in various kind of model systems has also been shown to signal through these intracellular mediators that are involved in the regulation of cell apoptosis, and some of the pro-apoptotic proteins are shown here to be stimulated by TGFβ in various kind of cell context-dependent manner. So this leads to the stimulation of the apoptosis, and this growth inhibition or cell cycle inhibition plus the pro-apoptotic activity of TGFβ then leads us to believe that TGFβ is really a tumor suppressive signaling pathway to inhibit the tumorigenesis. So indeed, when I was a postdoc in Dr. Michael Bratton's lab here, we found that the colon cancer cells, such as this cell line called HCT116, which is derived from patient predisposed to DNA replication error due to the mutation of DNA mismatch repair enzymes, often shows the loss of the type 2 receptor. So this is because that the type 2 receptor gene has this microsatellite kind of sequence of the 10A track here. And because of the loss of DNA mismatch repair function, and when the DNA gets replicated, very often a mutation is made, and then you lose an A here and becomes a 9A. Therefore, it creates a frame shift mutation, and then therefore we cannot detect this receptor anymore. So this leads to our discovery, as published in Science, that the type 2 receptor was a target of the loss of mismatch repair function. And on the other hand, if we re-express the Y-type type 2 receptor here into this cell line here, we show that the growth of the xenograft tumor is really significantly inhibited. So with that information, one would expect that in many types of tumors, the TGFβ receptors or other kind of pathway components will be mutated at a very high frequency. But in fact, this is not the case. So what we know now is that TGFβ's growth inhibitory activity is very often attenuated in the cancer. But these cancer cells still has an operational TGFβ signaling pathway. So shown here is the two cell lines. One is this MCF10A cell that is a non-tumorigenic immortalized human memory epithelial cell line, which, as you can see, that shows growth inhibition in a dose-dependent manner by TGFβ. On the other hand, human breast cancer cell line called MDA-MB231 cells, they are very resistant to TGFβ-induced growth inhibition, as shown here, but they still have an operational TGFβ signaling pathway. So in fact, while in certain type of cancer, which is believed to about 10% of all the cancers that harbors the loss of TGFβ signaling component, but many of the cancers that still retain the TGFβ signaling pathway, but with attenuated growth inhibition by TGFβ. So shown here again is a normal memory duct showing the phosphorylated Smad2 in the nucleus. And this phosphorylated Smad2 actually is more enhanced in this triple negative breast cancer. So at that time, this was about 20 years ago, many labs, including our lab, hypothesized that TGFβ signaling may be required for the survival and the malignant properties of some of the carcinomas, both in vitro and in vivo. So now we know that TGFβ can signaling through this autocrine as well as paracrine fashion, and to really promote cancer progression invading metastasis with various mechanisms as shown here. And these are just listed some of them. So autocrine signaling TGFβ has been shown to stimulate cancer cell survival and stimulate the epithelial-mesenchymal transition of the cancer cells, as well as to enrich the cancer stem cells. Signaling in the paracrine fashion, TGFβ has been shown to stimulate myofibroblast differentiation. Many growth factors in the tumor microenvironment is stimulated by TGFβ. TGFβ has been shown to be an angiogenic factor. Certainly stimulated the extracellular matrix remodeling. Some of the extracellular matrix proteases are regulated by TGFβ. And TGFβ, as we know, is also a very potent immune suppressor of the host immune surveillance, which unfortunately we have not studied much. But this is a large area of research here in the TGFβ field with respect to cancer immunology. So, as I said, about 2 decades ago, we don't have many TGFβ inhibitors. So my lab start to utilize the soluble TGFβ receptor as a potential TGFβ trap as well as an inhibitor. So the first receptor we utilized was this TGFβ type 3 receptor that I have not introduced to you. So this is also a cell surface protein here, and it is a proteoglycan, has 2 TGFβ binding sites per molecule here. And it binds to 3 TGFβ isoforms with a relatively high affinity. And so this is considered to be somewhat like an accessory receptor that will bind to TGFβ, and particularly TGFβ2 isoform. And because the TGFβ2 isoform has very low affinity for the type 2 receptor. So it binds to TGFβ type 3 receptor, then this type 3 receptor would then present TGFβ to the type 2 receptor, then and then recruit the type 1 receptor for TGFβ signaling. So the 2 binding sites on the type 2/3 receptor was used to call the E domain and U domain, as shown here. So if one can now then genetically engineer a stop codon at the extracellular domain, then one would get the soluble receptor here. So if we can express or use the soluble receptor as a kind of a therapeutic agent, then we supposed to be able to really use this receptor to trap TGFβ and prevent its signaling. So that's what we did in collaboration with Dr. Fernando Lopez Casillas. He is at the Mexico National University and generated this soluble type 3 receptor for us to use. So what we did was we used this recombinant soluble R3 and first inoculated tumor cells in nude mice. And then, so these are human tumor cells. And then when every tumor diameter is greater than 4 millimeter, then we will rank all the mice with various size of tumors, such that the 2 group, the control versus soluble R3 treatment group, the tumor median and mean are really kind of matched. So the first experiment we did was to actually inject the soluble R3 paratumorally. So about 50 microgram per mouse, we injected next to the tumor and showed that this injection or administration of the type 3 receptor can really inhibit the tumor growth, as shown here. We also tried this IP injection here at 100 microgram, excuse me, per mouse. And we also see a statistically significant inhibition of tumor growth. What I'm not going to show here is that we see actually a more significant inhibition of the metastasis of this human breast cancer cells to mouse lung, because we label the cell with the green fluorescent protein that we can image the lung to count for micrometastasis. But the next experiment we did was using this this colon cancer cell line that I showed you that has a mutation of type 2 receptor, therefore does not have any kind of TGF-beta signaling activity. So shown here is that this xenograft model is also can be inhibited by the systemic treatment with IP injection of the soluble type 3 receptor. So this tells us that the TGF-beta in this model system, because it has a mutation of the type 2 receptor, therefore it cannot signal induce the signal in the cancer cells, but yet the inhibition of TGF-beta inhibitor growth is this is the TGF-beta's paracrine role in terms of the work in the tumor microenvironment. And specifically, we show that this treatment of soluble receptor inhibited the tumor blood volume, indicating that there's inhibition of the angiogenesis here. So breast cancer, as we know, very frequently metastasized to the bone. And studies by a number of labs have shown that TGF-beta plays a major role in terms of driving the breast cancer metastasis to the bone. So we want to know whether we can either trap TGF-beta with this kind of soluble receptor, or use the TGF-beta type 1 receptor inhibitor, kinase inhibitor, to block this process. And at that time, the first TGF-beta type 1 receptor inhibitor was reported simultaneously by two groups, one from Eli Lilly as well as from Biogen. So we used this TGF-beta type 1 receptor kinase inhibitor and showed that it can dose-dependently, very potently inhibit the TGF-beta-induced the luciferase, the transcription of luciferase with this kind of reporter. The promoter region has this Smad binding element. So, and we also show that it is somewhat specific to the TGF-beta type 1 receptor called the ALK5. So these are all constitutive active type 1 receptor family members, ALK1/4. But 5 is more sensitive to this kinase inhibitor. So one experiment we did was that this human breast cancer cell model has been shown to induce the osteolytic bone metastasis when injected intracardiacally. And we also treated the mice with either the TGF-beta type 1 receptor kinase inhibitor or the recombinant soluble type 3 receptor for the 28 days. And when you look at this, the bone osteolytic lesions, we see that there's a clearly inhibition of this kind of lesions when we either treat it with a type 1 kinase inhibitor or the soluble type 3 receptor. So these again show that blocking TGF-beta signaling with either kinase inhibitor or the soluble receptor can really block the breast cancer-induced metastasis as well as the bone osteolysis. So we know, as I also said earlier, that this type 3 receptor has really high affinity for TGF-beta TGF-beta 2 isoform. On the other hand, the type 2 receptor has much higher affinity for binding to beta 1 and beta 3 isoforms. So we wanted to really test whether we can make a pan TGF-beta inhibitors by generating this kind of fusion receptor with the E domain of the beta glycan, which is type 3 receptor, and the soluble type 2 receptor here. And we used the bacterial expression system and then purified, generated this type 2 or type 3, type 2 receptor fusion receptor. We called it the BGR2. So excuse me. BGR2 was compared to the soluble type 1 type 2 receptor or soluble type 3 receptor, and by their activity of inhibiting the phosphorylation of Smad2 and Smad3, which can be stimulated by TGF-beta 1 shown here or TGF-beta 2 shown here. So as you can see, because the soluble type 2 receptor has high affinity for TGFβ1, then itself alone can really inhibit TGFβ stimulated phosphorylation of Smad2 and to some extent of Smad3 in a dose-dependent manner. On the other hand, the soluble type 3 receptor really does not have much activity here for the TGFβ1. And then if we use this fusion receptor BGR2, then we see a clear dose-dependent inhibition of TGFβ-induced Smad phosphorylation. On the other hand, the BG alone does not have much effect here. Similarly to the TGFβ2, type 2 receptor has very, very low affinity to TGFβ2, therefore you don't see much effect. Type 3 receptor is the one that binds to TGFβ2, therefore alone you can see the dose-dependent inhibition. Adding the type 2 receptor, soluble type 2 receptor here, with the BGR2 here, we also see a dose-dependent inhibition of this TGFβ2 activity. And interestingly, the half of this receptor BGE or the soluble R2 really do not show any effect. So combining together then showed the inhibition of the TGFβ2 stimulated the phosphorylation of Smad2 and Smad3. We also compared this fusion receptor to the PAN antibody or the FC-R2, which is a homodimer of the R2 connected by FC. And as you can see that we also show a really dose-dependent inhibition of the TGFβ stimulated luciferase activity in the comparing with the PAN antibody as well as the FC-R2. So so we show that this fusion receptor is really more effective. So utilizing this one, we want to know whether it is therapeutically active. So we used another model, which is a prostate cancer cell line we developed here on our campus here by another lab. And this cell line actually induces extensive, bone growth. So very often the prostate cancer cells can induce this osteoblastic lesions with increased bone growth. So, and we treated the mice with this type 1 kinase inhibitor or the soluble receptor we generated here. After injecting this cell line intratuberally here, that shows the formation of the tumor here. So this is by bioluminescence imaging here. We show that the treatment with either of the TGFβ inhibitors really inhibited the growth of this tumor in the tibia. And interestingly, these tumors were also metastasized to the lung. And we using this fluorescent imaging because the cell lines is also express the GFP. So we see that this TGFβ inhibitors can really inhibit the lung metastasis, and particularly with this fusion receptor. So this is the early days of our development of TGFβ soluble receptor as inhibitors. Now I'm going to switch the gear to tell you a story that we published recently in the clinical cancer research with respect to how TGFβ signaling is activated by chemotherapeutic agent. So we know that chemotherapeutic agent has been shown to stimulate TGFβ signaling mainly in breast cancer model. So when Dr. Haiyan Zhu joined my lab as a postdoc, she wanted to know, was interested in the OBGYN type of cancer. So we analyzed this publicly available data published by Moreno et al. So this data contains the RNA expression data from the ovarian cancer tumor of 24 patients. And that also has 9 untreated patients with the tumor. So we analyze this RNA expression data and generate this heat map showing the differentially expressed genes after treatment with the chemotherapeutic agent here. So as you can see, most of the patients except these two class together in terms of gene expression. And so this red bar shows the upregulated genes after chemotherapy treatment, and these are showing the downregulated genes after chemotherapy. So and if we look at these differentially expressed genes with respect to what kind of biological processes they participate, we see many of the cell cycle functions here that are very low in terms of the adjusted p-value FDR. So indicating that the chemotherapy agent really regulates the many genes that are involved in cell cycle, which is really expected. And we then also look at these differentially expressed genes as to what are the upstream regulator of these genes. And the top one, which has this activation Z-score as a positive value, indicating that the Z-score positive indicating this pathway or this upstream regulator is really activated. So the top one with the least p-value is the p53, which is also expected because the chemotherapeutic agent very often, activated the p53 pathway by inducing the DNA damage. So the next one, which is positively activated, is this TGFβ1. So this indicated that the chemotherapeutic agent really can regulate many gene expression that are downstream target of TGFβ1. So this is in ovarian cancer. Dr. Su also used the pre and post treatment with the chemotherapeutic agent, the cisplatin, for the cervical cancer set of the tumors, and also found that after this cisplatin treatment, she sees an increased nuclear localization of the phospho-Smad2. So this is total Smad2, which is not significantly different, but the phospho-Smad2 is significantly increased after the treatment. So these lead us to hypothesize the chemotherapeutic agent may activate TGFβ pathway, resulting in chemoresistance in ovarian and cervical cancer cells. So our question was how this chemotherapeutic agent activates TGFβ signaling, and can we really use TGFβ traps to block TGFβ signaling to enhance the efficacy of the chemotherapeutic agent? So during this whole period, our approach is to use this 4 chemotherapeutic agents as listed here, and 4 cell lines, 2 from cervical cancer and 2 from ovarian cancer here. And we used a novel TGFβ trap here that we designed it. And so this, first of all, I want to show you the data that the chemotherapeutic agent here really showed the stimulation of phospho-Smad2 and Smad3 here, as you can see here for all the 4 cell lines here in various degrees, obviously. In some cells that the cisplatin may be more effective, and in others maybe paclitaxel. So these are the 4 drugs with the concentration we used it, and we see basically more or less that we see a stimulation in a time-dependent manner by these chemotherapeutic agents in these various cells with respect to the Smad2 or Smad3 phosphorylation. So TGFβ, we found that these chemotherapeutic agents actually can stimulate the TGFβ transcription by these 3, not a paclitaxel here, and also show TGFβ2 transcription was significantly stimulated by Dr. Rubinson here. More importantly, TGFβ1 protein, either the total or the active form, as TGFβ is secreted in latent form, so we are more interested in the active form, can be increased by these 4 drugs in the cell-conditioned media. So clearly, TGFβ Smad phosphorylation is apparently due to the increased active TGβ stimulated by these kind of chemotherapeutic agent. So we now wanted to test a novel TGβ trap here. So this one is in collaboration with Dr. Andrew Hink, who is really an expert in studying TGβ as well as receptor structure. So this is really from him. He found from many years of study that the type 3 receptor, this E domain type 3 receptor, really bind to this homodimeric active TGβ in this middle region here, but the type 2 receptor binds to where these two end here. Okay, so if we use this previous TGβ fusion protein called BGR2, now it's labeled as ER. This will bind to the this part of TGβ, but it will expose this end of this this one of the homodimer that is still available to bind to TGβ. So that in this case, it can bind to the cell surface type 2 receptor, and then it could induce the signaling pathway. So Dr. Hink, hypothesized that if we add another R2 here on this end, then therefore this R2 BGR2, we call it abbreviated as RER, then will really block all the binding sites of TGβ to the receptor. Therefore, it really will be more potent. And indeed, this is the case. TGβ, when it stimulated the SMAD 3 SMAD 2 phosphorylation, can be inhibited by this ER in a dose-dependent manner, but it's more potent if we use this RER, as you can see here, that at 0.1 nanomolar, it already showed the inhibition here. So we further tested this RER and comparing with the BGR2 or ER, as shown here, in terms of blocking the TGβ2 or TGβ3 stimulated this reporter system here. And in both cases, that this RER is more, kind of active or potent than the ER, showing in both cases here. So going back to the ovarian and cervical cancer, we can show that TGβ stimulated the phosphorylation of the SMAD2 and SMAD3, and RER itself mildly reduced this phosphorylation at the baseline, but really inhibited the TGβ-induced the SMAD2, SMAD3 phosphorylation, as shown here when we combine beta-1 with RER. And this is also shown to be inhibited by this HTAS, which is a type 1 receptor kinase inhibitor. So we used both inhibitors throughout this experiment in these 4 cell lines, and show that in the case of cisplatin, it really inhibits in all the cases here except this cell line. And paclitaxel, we also see an inhibition in some cell lines here by the 2 inhibitors. And the same thing is true for the other 2 chemotherapeutic agents here, that we see some inhibition when we combine the chemo drug with RER or the HTS in these various cell lines, as you can see here. So similarly for this camptothecin, that is also the case. So basically, we also showed that because TGβ has been shown to stimulate the epithelial-mesenchymal transition, we also see that these chemotherapeutic agents, in this case the cisplatin, would stimulate, like TGβ, of the snail, which is the EMT marker, and also TGβ to some extent inhibited E-cadherin, and then this it can be reversed when we add the cisplatin with the RER or this kinase inhibitor here. So here we show that EMT is associated with increased migration. So TGβ can potently stimulate the tumor cell migration through this kind of transwell format. And so do these chemotherapeutic agents in this case, at least the 3 drugs, not the paclitaxel here, can stimulate the cell migration here in these 2 cell lines. And if we add this IER to inhibit and the IER or the HTS really can inhibit this cisplatin-induced cell migration here in HeLa cells, and also can inhibit the doxorubicin-stimulated migration also in the HeLa cells, as you can see here. So another function of TGF-beta signaling pathway is to really stimulate the cancer stem cell or enrich the cancer stem cells. And these cancer stem cells can be really identified by looking at some of the cancer stem cell markers. Here is the CD44, CD133, as well as this ABC transporter. So these are the cisplatin is shown to stimulate the CD44 expression, and that is moderately inhibited by the IER. That is in the at the mRNA level, and also shown here is the inhibition by the IER here, as well as by the kinase inhibitor here. Similarly here. So, and we also can use the flow cytometry to really score the CD44, CD133 positive cells here. These are supposed to be the cancer stem cells here. And then the treatment with cisplatin really increase this population here, which can then be really inhibited by the addition of this, the IER. So all these data indicate that this IER is quite potent in terms of blocking these chemotherapeutic agent-induced TGF-beta signaling. So finally, we used a xenograft model of this omega-3 ovarian cancer cell line. And so this is immune deficient newt mice. Showing here is that the cell will form the tumors that will steadily grow. And IER alone really did not show much effect in this model system. So we also used the 2 doses of the cisplatin. One is a 2.5 milligram per kilogram. Another one is 5 milligram per kilogram. And then without or with the IER. So at a low dose, it inhibits the xenograft growth, but the addition of the IER really inhibited this xenograft growth quite significantly, as shown here. At the high dose, the cisplatin itself was very potently inhibiting the tumor growth. Therefore, we do not see a statistical significance. At the end, weighing the tumor volume here, we see a steady decrease of tumor volume here, even though this one, comparison of this one, did not show a statistical significant difference, but it only showed a trend here. And the tumor volume really showed a growth inhibition. So analyzing these xenograft tumors, we also see that the treatment of the cisplatin really stimulated the TGF-beta mRNA level, which is somewhat reduced. But if we look at the active TGF-beta-1 in the tumor extract, we really see that the cisplatin treatment stimulated the active TGF-beta, which can then be reduced by the combination treatment with the IER. And this is also shown here that the cisplatin stimulated phosphorylation in the tumor extract, and this is reduced. And this is the quantification of the phosphorylation divided by total SMAD2 ratio here, showing that the stimulation of the phosphorylation, which can be reduced by the combined treatment with the IER. Similarly, for looking at some of the EMT or stem cell markers in the tumor, we also see a stimulation of the SNAIL2, and then which can be reduced. And so is this CD33. We can see a reduction here, CD44 as well, and then ABCG. Okay, so all these indicate that the chemotherapeutic agent here really, even though it can inhibit tumor progression, but unfortunately, it has this unwanted side effect of stimulating TGFβ signaling. And TGFβ, as we know, would stimulate the EMT as well as the cancer stem cells that leads to chemo resistance, and also can increase the cell survival and leading to metastasis. So what we are showing is that if we can use this kind of TGFβ trap to really block this pathway, that should enhance this chemotherapeutic agent. So in conclusion, that we have found that the systemic treatment with the soluble type 3 receptor reduce the tumor burden, angiogenesis, metastasis instance, as well as osteolysis. And this fusion receptor we call the BGR2 appears to be more effective than pan-TGFβ inhibitors, such as the pan-TGFβ antibody. So we also show that chemotherapeutic agents can activate TGFβ signaling by stimulating TGFβ1 production and secretion, resulting in EMT as well as cancer stem cell enrichment and increased cancer cell migration in both human ovarian as well as cervical cancer cells. And then combination with a novel TGFβ trap we call the IER with this cisplatin really neutralize cisplatin stimulate TGFβ tumor-promoting activity, leading to a more efficacious inhibition of the ovarian cancer cell growth in a xenograft model. So with that, I like to conclude by acknowledging many of the lab members who really contributed to this line of the research in the lab over the years. So Dr. Abika Bendio-Padaha, who is not in this picture, initially started the soluble type 3 receptor work. And the BGR2 was really made available by this graduate student in the lab who expressed this BGR2 in a bacterial system and then refolded to make it active as a TGFβ trap. I talk about Haiyan Zhu's work. She is in the picture here in collaboration with Xianggu, who did the bioinformatic kind of analysis of the genes that differentially express in ovarian cancer with without chemotherapeutic agent treatment. And then Summer is here, helped Haiyan to some extent, with also additional help from Joan and Hakim here. And our collaborators include Dr. Andrew Hink, who is currently now at the University of Pittsburgh, Dr. Joanne Zhu from the Medical College of the Windsor Medical University, and I mentioned Dr. Fernando Lopez Casillas from UNAM in Mexico. So the funding sources are mostly from NCI, with some help from our cancer center, it's called the Mace Cancer Center at our university here. So with that, I wish to thank everyone to listen to my talk, and I would be happy to address any of your questions. Thank you.
Thank you for that wonderful presentation, Dr. Sun. And as a reminder to our audience, as we enter our Q&A session, you may ask questions at any time using the Q&A tab. Our first question concerns I want to ask about how the chemotherapeutic agents induce TGF-beta activation. And it seems like there are varying levels depending on the agent, because different agents had different levels of TGF-beta, Smad phosphorylation, and different levels of susceptibility to blockade. Do you have any ideas or hypotheses with regard to mechanisms and why and how different agents affect TGF-beta signaling differently?
Right. So this is really a good question that we have been asking ourselves. Unfortunately, after Dr. Haiyan Zhu finished her postdoc training in my lab, we have not really looked into this more deeply. So what we know is that it probably different agents certainly has different potency, as this question, the person who asked this question pointed out. It appears to be that this may be at different levels, including, as I showed, some at the mRNA level in terms of transcription. We know that these chemotherapy agents, while they mainly damaging DNA or interfere with the microtubules, they very often can regulate various signaling pathways. So some studies have really shown, such as paclitaxel, can stabilize macrotubules, and the TGF-beta pathway to some extent is kind of dependent on these macrotubules to transport the Smad proteins from the near cell surface to the nucleus. So, TGF-beta-1 particularly, its transcription is auto-regulated. So if you enhance TGF-beta signaling, such as this mechanism of macrotubule, then one can, basically hypothesize that the stabilization macrotubule enhances the TGF-beta signaling pathway through the transport of the Smad proteins, will then stimulate TGF-beta-1 transcription. To some extent, and this has been shown not only by our lab, but by Dr. Carlos Artiega's lab, also in breast cancer model. On the other hand, some of the chemotherapeutic agents may more specifically activate the TGF-beta by some kind of mechanism, maybe related to the expression of the extracellular proteases that are involved in TGF-beta activation, because we see a more kind of uniform increase of active TGF-beta-1 in the cell-conditioned medium, as well as in the xenograft tumor, after treatment with various chemotherapeutic agent. So this is likely to be at the multiple levels, but we really have not studied that. So I don't have a direct answer to that, and these are basically my speculation.
Thank you, Dr. Sun. Next question. We know that TGF-beta can operate as both a growth inhibitor and as a growth promoter. What determines whether TGF-beta functions as a suppressor or a promoter? And in the course of this talk, we talked a lot about the promotion side, but have you ever seen evidence of a TGF-beta mediated inhibition mechanism coming into play during your work?
Yes. So this is, again, a very, very insightful question. So my early career, starting from my postdoc, as I showed that we, as TGF-beta was cloned, I think around 1985, and many of the first studies published was showing that TGF-beta is really a potent growth inhibitor, and particularly in epithelial cells and lymphocytes. And then later on, mechanistic insight, was also described, and showing that the CDK inhibitors, the cyclin-dependent kinase inhibitors, are transcriptionally inhibited by TGF-beta signaling. On the other hand, some of the growth promoters, as I showed in one of my slides, such as the c-Myc, is really transcriptionally inhibited by TGF-beta pathway, and these all contributed to the role of TGF-beta in terms of the blocking the tumor progression. So we also have published a study, many years ago, right after I became independent, showing that if we express the type 3 receptor, Y-type type 3 receptor, which is membrane anchored and which can facilitate TGF-beta signaling, in some model systems, we show that this would enhance overexpression of the Y-type type 3 receptor will enhance TGF-beta signaling, leading to the inhibition of the cell proliferation, as well as inhibition of their growth in vivo in a xenograft setting. Early on, studies from Dr. Michael Bratton's lab, who is my postdoc mentor, by many of his students, also showed a number of studies that we overexpressed type 2 or type 1 receptor in the colon cancer cell lines that show either mutation of the type 2 receptor or the downregulation of type 1 receptor. Also, if we overexpress these receptors, then it will inhibit cell proliferation very potently and inhibit their tumor formation as well. So there's early studies, many of them really show the tumor suppressive side of the function. Now, with respect to why TGF-beta, basically, I believe it's the current consensus that as tumor formed and progressed to a more advanced stage, then TGF-beta, growth inhibition or tumor suppressive function is really lost, and that is also due to many of the kind of mechanisms, such as loss of TGF-beta stimulation, stimulation of these CDK inhibitors, and loss of the inhibition of these growth promoters such as c-Myc, because c-Myc is very often, the gene is amplified in various kind of cancers. Therefore, it no longer responds to TGF-beta inhibition. Loss of some of the downstream signaling pathways such as Smad proteins, and particularly Smad4 in certain type of cancers, mainly in pancreatic cancer. So all these leads to TGF-beta now does not signal through these kind of inhibitory function, but yet, as I also showed in one of my slides, the TGF-beta can stimulate some of the MAP kinase pathways, as well as the PI3 AKT pathways, and that could contribute to TGF-beta regulation in terms of tumor promotion. Certainly, Smad pathway is still, in some of the later stage models, is still there, and in fact, it's been shown to mediate this EMT, and we know the EMT is also a sign of the progression and become more aggressive. So this is, I guess, the balance between TGF-beta inhibition and promotion with so many genes that TGF-beta regulate.
Thank you, Dr. Sun. Next question. Have you observed any side effects to TGF-beta trapping in vivo over a prolonged period of time? And this question is relevant because TGF-beta does have so many other different functions across different systems. But the question specifically wanted to inquire whether you saw anything that would affect immune development and immune system regulation.
Yeah, so this, again, another question, a very good question. We, as I pointed out, unfortunately have not studied the immune part. In our xenograft models, we always use immune deficient mice because we wanted to study human cancer cell lines that will only form tumors in the immune deficient mouse model. So in those cases, as long as we treat the mice with daily injection of the soluble receptors, we have not seen really kind of adverse effect in terms of mouse behavior or the weight change. So that does not really say that it has no kind of side effect, which is something that I think needs a really kind of deep investigation. The hypothesis that I have is that while we, the person who asks this question probably knows that the kinase inhibitors of TGF-beta type 1, type 2 receptor has been shown to elicit various type of quite severe side effect. Therefore, they are not used clinically. There's clinical trials, but I don't think it has been FDA approved for the human use. This is because these are small molecules, and they certainly get inside the cell. In addition to their maybe targeted kind of non-specific effect on various other targets, because these are basically ATP competitive binders. They certainly can block very severe TGF-β signaling pathway in both tumor cells as well as normal cells. On the other hand, these soluble receptors, they don't get into the cell. They basically kind of really mop up the extra TGF-β that is very often upregulated in the tumor microenvironment. So the hypothesis is that the soluble receptors don't have this kind of really potent and severe inhibition of TGF-β as the TGF-β small TGF-β inhibitors that inhibits the kinase of the receptors. So this obviously needs to be further tested. But some studies, I think, I don't know at what stage, but the soluble type 2 receptor FC kind of structure or the proteins, I believe they are moving forward with clinical trial. So let's see what happens.
I suppose related to that question, do you have any data on how well these soluble receptors penetrate tumors, reach sites of tumorigenesis, versus how long they stay in the circulation or maybe stay in non-target organs?
Yeah, so again, good question. So we know when we do the IP injection, we can detect this TGF-β fusion receptors in circulation in the blood. I mean, after about 30 minutes. And they are short-lived. So in circulation, we can detect them early after injection, but then they will go below the detection level of our ELISA assay. We can, I think in some of our early studies about soluble type 3 receptor, we can detect the receptor in the tumor tissue by, again, ELISA. So they, I think they are cleared by, through the kidney and maybe also degraded to some extent, even though soluble type 3 receptor naturally is detectable in circulation because this receptor is basically shed and then get into circulation. So it's believed that soluble type 3 receptor is somewhat more stable. So we know when we inject IP, we have labeled the soluble fusion receptor, this BGR2, with a fluorescent dye, and they often ended mostly in the liver and some in the urine through kidney. So, this is what we know about this receptor, and therefore, what we see is if we do the daily injection, and some of the studies we do every other day, we see an efficacy. But I don't think the efficacy will stay that long if we do once a week injection, because we just don't see them to stay very long in circulation.
Thank you, Dr. Sun. We have time for one final question, and that would be, what is the status of the RER or TGF-beta traps in general with regard to clinical application?
Yeah, so this RER, we have a US patent. Well, it's US and Europe, I think. We have some investors interested in looking this protein before. Currently, it is with our technology development office of our university. And if someone is interested in talking to these folks in our Office of Commercialization, I can certainly provide the contact information. So to be honest with you, I don't know whether this is currently licensed out or not, because I know that there was a few companies interested in licensing this IP for further development.
Thank you, Dr. Sun. Unfortunately, that is all the time that we have for today. If you have any further questions, please consider reaching out to the speaker directly. His email is shown on the screen. As a reminder, this webinar will be archived on the scientist's website, and you will receive an email notifying you of when the on-demand webinar becomes available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientist, I would like to thank our speaker, Dr. LuZhe Sun, and as well as our sponsors, Sinobiological, Nanotemper Technologies, and 10x Genomics. Thank you, everyone, and have a pleasant day.